Hello, and welcome to membrane permeability modeling, the 2-parameter and 3-parameter formalisms. My name is Nick Zuchowicz with the Department of Mechanical Engineering at the University of Minnesota. What are we going to talk about today? First, motivation. Why does cryopreservation matter, and why should we do membrane permeability modeling? We'll move into the 2-parameter formalism, and we'll finish up with the Kedem-Kachalski 3-parameter formalism for modeling such systems. A follow-on video will look at experimental estimation of membrane parameters, curve fitting, and examples of applications for these models. First, let's look at some history. The first intentional cryopreservation succeeded in 1938. This was the cryopreservation of human sperm in a thin film of liquid on a glass slide plunged into liquid nitrogen. A few years later, the first cryoprotective agents, CPAs, were identified. CPAs are chemical additives that remove water from cells, or permeate into the cells, or both, and enhance survival through cryopreservation. A number of other CPAs have since been discovered. Some of the most commonly used are dimethyl sulfoxide, propylene glycol, ethylene glycol, and trehalose. Cryopreservation is already widely applied in human reproductive health. Sperm, eggs, and embryos can be stored for years or decades. Looking to the future, if human organs can be preserved whole in cryogenic storage, this will support better matching of donors to recipients across great distances and across time, and will reduce the wasteful discard of usable organs. There are important applications in biomedical research for cryopreservation. For example, the Zebrafish International Resource Center houses both a live bank of fish and a frozen sperm bank representing tens of thousands of alleles, created by research groups around the world. The sperm can be thawed and used to reconstitute live lines, then the juvenile fish, carrying known alleles, can be shipped out to researchers on demand. This is hugely more efficient than maintaining the bank's full diversity in vivo. Cryopreservation is a valuable suite of technologies, enabled by the use of CPAs. We want to model how cells behave when exposed to CPAs, so as to improve our cryopreservation approaches. By way of example, on the left we have sea urchin eggs, exposed to a non-permeating CPA, trehalose. The eggs shrink and stay shrunk. On the right, we have eggs of the same species exposed to a permeating CPA. The eggs shrink, but then recover their volume over time. The questions are, how do we model these behaviors, and how do we get information from such experiments? We'll start our investigation of modeling with the 2-parameter formalism. This formalism comprises a pair of simultaneous ordinary differential equations, ODEs. In the first equation, the first term is the rate at which water is lost or gained by the cell. This rate is the product of L sub P, the experimentally derived hydraulic conductivity, the membrane area of the outside of the cell, the gas constant, the temperature of the system, and the difference between external osmolality, superscripted E, outside the cell, and internal osmolality, superscripted I. In the second equation, we have the rate at which moles of permeating solute, N sub S, enter or leave the cell through the membrane. This is the product of P sub S, the solute permeability, the membrane area of the cell again, and a difference of external and internal osmolalities, but here the sub S tells us that we're looking only for the osmotic pressure produced by the permeating solute. So what is hydraulic conductivity? Let's look at the units. We have microns per minute per atmosphere. We can multiply top and bottom by microns squared to find some intuition for what the units mean. We can see this as some volume, microns cubed, of water, passing through some membrane, microns squared, per minute, per atmosphere of osmotic pressure across the membrane. Similarly for the units of solute permeability. We can multiply top and bottom by centimeters squared, giving us a similar intuition. In much of the cryobiological literature, solute permeability uses centimeters, while hydraulic conductivity uses microns. When coding up these models, however, we have to be careful to use uniform units to get a sensible answer. Both hydraulic conductivity and solute permeability are functions of temperature, each with its own activation energy, E sub A, that represents how the parameter scales with temperature. This is important in cryopreservation because as a cell is cooled, the rates at which water and solute cross the membrane will vary. Let's look briefly at the units of osmolality. Molarity will probably be familiar, since that's often how we make up solutions in the lab. However, in these models, we use osmolality, dissociated moles of solute per kilogram of solvent. Let's plot the normalized cell volumes over time predicted by these equations. Here we'll introduce one more concept, the bound fraction, the part of the cell's volume that does not participate in the osmotic behavior of the cell. For this discussion, we're taking it to be 25%, 0.25. When we model a cell exposed to a non-permeating solute, that is, the solute permeability is 0, the cell will shrink and stay shrunk. As water is expelled, the internal osmolality of the cell rises to match the osmolality of the cell's surroundings. With a non-zero solute permeability, there will first be that expulsion of water, but then there will be a reswell as the solute enters the cell and brings water back in with it. With high solute permeability, the initial volume of water loss is less. Solute can get into the cell more quickly, reducing the osmotic pressure across the membrane. And with a very high solute permeability, almost no water is lost at all. Let's play with a couple of other parameters. Holding solute permeability constant, but varying the hydraulic conductivity, we see that the initial water loss is affected, but the final volume of the cell does not change much. On the other hand, varying the external permeating solute osmolality does vary the final volume achieved. Now we'll move on to the Kedem-Kochalski 3-parameter formalism. As with the 2-parameter formalism, we have two simultaneous ODEs, but there are some differences. First, notice that the first equation represents the rate of loss or gain of water and solute volume put together, not just the water. Second, we still have differences of osmolality, but we've split apart the non-permeating solute, denoted sub N, from the permeating solute, sub S. We still have hydraulic conductivity and solute permeability, but we also have this new parameter, the reflection coefficient, sigma. This quantifies the degree to which permeating solute and water interfere with each other's passage through the same membrane. Sigma is constrained to be between 0 and 1, and in fact cannot be greater than the expression on the right. It's equal to 1 only in the uninteresting case where the solute permeability is 0. If there's no interaction between solute and water, then sigma exactly equals the expression on the right. As interaction at the membrane increases, sigma decreases. And sigma, like the other parameters, is a function of temperature. Why would we choose one model over the other? Where possible, we should choose the simplest available model that adequately represents the system of interest. Fitting the 3-parameter model where it isn't suitable gives more freedom in the parameters, which can give a spurious fit. Here we have some cases to guide us. In each plot, solid lines represent 2-parameter solutions, while dotted lines represent the corresponding 3-parameter solutions for the same inputs. In the top plot, with no solute-water interaction at the membrane, and low internal and external osmolalities, the 2-parameter and 3-parameter solutions are nearly indistinguishable. Here, the simpler 2-parameter model is the best choice. However, when all else is held constant and a solute-water interaction is introduced, that is, sigma decreases, the 2-parameter model deviates more and more from the correct solution. The 3-parameter solution is best. Finally, with high external permeating solute osmolality, we're violating the dilute assumption that is built into the 3-parameter model. The 2-parameter solution is best here. Efficiently finding and improving cryopreservation protocols depends in part on models that predict how cryoprotective agents traverse the cell membrane. The 2-parameter and 3-parameter solutions are related models, each with its application. A follow-on video will discuss how these equations are applied experimentally and how they can be used to improve a cryopreservation protocol. Thank you for your attention.